Hi, my name is Paul Neum. I'm a professor at the University of Washington and at the Fred Hutchinson Cancer Research Center in Seattle. I'm going to talk to you today about how ATR functions in the normal cell cycle and how we now believe that it may be able to help us kill cancer cells when we use an ATR inhibitor. The concepts that I'm going to show you today have withstood the test of time over a number of years. I made this animation using Flash, which is no longer a working platform. So I'm converting it now into a short animated video summarizing the biology here. And while some of the concepts are, each individual concept is simple, how it goes together is not intuitive. And that's why I'm putting together this video. Happily, we are now in the year 2021, really moving into clinical trials after many years of developing potent and selective small molecules by pharmaceutical companies, which will allow us to see if this is really going to work. And the exciting thing that we've learned just in the last few years, more recent than what I'm going to summarize today, is that using an ATR inhibitor like this seems to cause what's called immunogenic cell death, or make the cancer more visible to the immune system, which is an exciting concept. So let me share this animation with you now. This is what we made several years ago, and mostly it says this is how a normal cell, this is a nucleus, when it replicates, normally goes into a mitosis and looks like this. And ATR's job is to sense trouble spots in DNA copying or DNA replication and block this process, which is when the cell tries to divide when it is only partially done with copying its DNA. So let's get started here with reviewing what is a normal mitosis. So in the normal cell division cycle, DNA replication occurs here in S phase, and a complete copy of the genome has to be made prior to mitosis or division over here. Normal mitosis occurs when replication is complete prior to chromatin condensation, the initial phase of mitosis, when all the DNA gets packed into these type things that we recognize as chromosomes. So in the normal simplified version of the cell cycle, G0, the resting phase, leads to the initial G1 phase and into S phase where DNA is replicated. And that gives us this thing that looks like a full, I'll just show you this one more time, a normal mitosis here with chromosomes that we can recognize. What can happen when that doesn't go right is something called premature chromatin condensation, where essentially before all the DNA is completed replicating, the cell actually biochemically enters mitosis and tries to condense its chromatin, and you get these giant mess of what looks like pulverized DNA here, where short segments and long segments have been copied, and it's a tangled giant mess, and that leads to cell death. And this is what happens if there's extensive incomplete replication and ATR is not functional. So how does ATR normally recognize trouble spots in DNA replication? So on the left and on the right are things that can cause cellular replication stress. On the left are forms of DNA damage, like chemotherapy or ultraviolet or ionizing radiation. And on the right are small molecules that can inhibit polymerases and such. Doesn't really matter. Any of these can cause a stall in a replication fork, or the area where the DNA is actively being replicated. And if you have a stalled replication fork, what's going to happen is you'll have a bunch of extended single-stranded DNA here, which binds to a protein called replication protein A. And normally replication protein A won't really bind to DNA because you don't have single-stranded segments of DNA sitting around normally for a prolonged period. But when you've got trouble at one of these replication factories, then the RPA comes in, and that attracts a complex containing ATR and the ATR interacting protein that recognizes this. And as that aggregates in these trouble zones, the complex stabilizes this replication fork and prevents disastrous double-strand breaks, and also blocks new areas of the genome from starting to replicate. There are usually hundreds of little individual zones that replicate at different times in S phase, and this process then, by phosphorylating key targets such as CHEK1 and the downstream signaling pathway, will prevent mitosis from proceeding normally if there is a problem zone. If ATR's function is inhibited, even if these problem zones exist, the cell can no longer sense those. And that's the key role of ATR, is to sense that replication is not complete. There's a bunch of single-stranded DNA sitting around that shouldn't be sitting around, and then it sends these safety signals. But if ATR is inhibited, then mitosis actually proceeds with these problem zones, and you either get premature chromatin condensation, which I showed you, which is massive problems as you enter mitosis, or you get individual chromosomal gaps and breaks, which are survivable and are sort of hallmarks of cancer cells, indicating that this pathway isn't always completely working optimally. Now let's put these two things together. We showed you before the biology of the cell cycle and now how ATR works in the individual zones of chromosomes. And in a normal cell, first, with no damage, the polymerase replicates at the genome at a normal rate, and there's really minimal role for ATR. There's not a lot of problems as you're completing replication, and each one of these is zipping through here. The green strands are the new copies of the DNA, and you go through the cell cycle and you have a normal appearing mitosis. In contrast, if you have damage in a normal cell, then you get a number of spots where the DNA is not readily copyable due to the damage. And a normal cell has all these quality control mechanisms. First of all, the retinoblastoma protein, for example, regulates entry into the active part of the cell cycle. And then P53, P21, and many other damage sensing proteins get turned on in a damage sensitive way and provide time for the cell to carry out repair. And so a lot of these damage sites go away based on that extra time, but some may remain. And then we have a role for ATR that comes in, senses this area where replication is not proceeding normally, and provides extra time for repair to happen. And stabilizes things, and then the cell largely can complete its replication without significant problems. That's how things normally function for ATR with a little bit of damage on board. Now we're going to take a cancer cell with some DNA damage, and the same damage is induced in the cell based on ultraviolet or whatever ionizing radiation. And then, as compared to a normal cell, cancer cells are sloppy in entering the cell cycle. That's why they're cancer cells. They divide a lot, and then they don't have time to repair, and they don't have this induced sensor of the damage itself, so for example, downstream of P53. So the cancer cell enters S phase with a lot of damage on board, okay? So all of these, 3 mutations, for example, here in each of these 3 segments are present, but that's okay. Lots of ATR comes in, and a very strong ATR signal is induced, and this leads to a lot of repair happening and minimal damage. There's a little bit of premature chromatin condensation or some gaps and breaks. Some cells die, but most are okay and can proceed, having had heavy help from ATR. Now we're going to put all this together. We've got a cancer cell with DNA damage and the ATR inhibitor. So the same amount of damage as usual, but the cancer cell lacks these early and mid cell cycle controls here. So again, we enter S phase with lots of damage on board. There's plenty of ATR to come in and sense this problem here and bind. However, we throw in a small molecule. In this case, this was caffeine, the first ATR inhibitor that was identified, not selective or specific, but definitely could induce this process. And extensive premature chromatin condensation occurs and cells die a p53 independent cell death that we now believe is immunogenic. And that is a very exciting thing if we combine immune therapy with this kind of logical inhibition of cell cycle checkpoint basically in cancer cells. So in summary, ATR is a protein kinase whose job is to prevent chromatin condensation, the first stage of mitosis, from beginning if the genome has not fully replicated. And it works by binding stalled or incomplete replication sites and then phosphorylating critical targets to block mitosis. In the absence of rapid cell division or DNA damage, like in normal cells, ATR is less critical and doesn't really cause too many problems if we partially or moderately inhibit it. However, in cancer cells that are deficient in G0, G1 checkpoints, ATR is really important for inducing this replication checkpoint, and such cells begin DNA synthesis then with extensive unrepaired damage due to the lack of time spent in G0, G1 for repair, and an ATR inhibitor can selectively kill such cells, especially when combined with a little bit of DNA damage. And we now know that that leads to, at least in certain circumstances, a immunogenic cell death that pulls in T cells and can help you fight the cancer. So we will now, in the early 2020s, be embarking on a number of trials that we're directly or indirectly involved in, and it'll be very exciting to see how all of these concepts come together to hopefully help patients. Thank you very much.